everyone, welcome to another special topics lesson here in Chemistry 2. Today our topic kind of falls within the realm of biochemistry. Bio meaning life, and so what we're typically talking about within that realm is kind of the chemical processes that occur within living things. And so one of the standards that Chemistry 2 kind of had set out for it was to be able to kind of compare and contrast some different structural kind of significances of different biochemicals, kind of the four main ones being carbohydrates, nucleic acids, proteins, and lipids, and kind of talk a little bit about how their structure affects, what they do as their function, in living organisms. And for me, this was kind of stuff, I guess I would have done junior year of college, and there's a lot more to this. It was broken up into a Biochemistry 1 and Biochemistry 2. Had a great teacher, though, who's really passionate, about the subject and did a really good job teaching it. So it's an interesting course. I definitely recommend if you want to learn more about how chemistry applies to, just kind of how we, function, some of the different processes that go on, like within our bodies. And so what we're going to do today is kind of do a brief overview of the four main macromolecules that play a big role within biochemical processes. The four main macromolecules are proteins, lipids, carbohydrates, and then DNA and RNA are kind of in their own category. And so these macromolecules, they consist of kind of repeating things called monomers. And so the monomers, mono meaning one, those are kind of the individual units that when you put them together, you generate what's called a polymer. Poly meaning many. And so many of the monomers together make up the polymers, which then are your proteins, lipids, carbohydrates, DNA, RNA, etc. And so before we get to looking at our chart, let's take just a little bit of time to kind of examine the monomers, or like the basic kind of puzzle pieces that make up each of these macromolecules. And what we'll be able to do, kind of with now our background of being able to look at molecules, is we'll be able to identify some of their structural characteristics, and then we'll try to put those in place with how they function. So amino acids, you guys had kind of an article to read on this the other day. What you may notice is you have kind of an amine group here at the end, and you also have your carboxylic acids. So you've got kind of two functional groups there that can do different things. If you'll notice kind of that nitrogen and hydrogen, that kind of is a characteristic where we could have some hydrogen bonding, and as well as that OH group, that's something that could participate in hydrogen bonding as well. You're also going to have some regions of more electron density, with your oxygens and your nitrogens being more electronegative. And so that's kind of your typical structure of your amino acids, which are your monomers for different proteins. Then for fatty acids, you'll see, again, they have in common with an amino acid, they have that carboxylic acid group, okay, so that carbonyl attached to an OH. But then what this little N means is that means you can have many of those segments that repeat. And so the rest of a fatty acid is just kind of a big, long, kind of carbon and hydrogen system. Sometimes you'll see double bonds, sometimes you'll see triple bonds, different things like that, but it's a big, long, chain of lots of carbons and hydrogens that are attached to that carboxylic acid group. And so fatty acids are your monomer for lipids, all right? And just by the picture, lipids kind of may start to remind you of like your cell membranes. It's like we talked about phospholipid bilayer, kind of when we made the butter earlier. So that's where that comes into play. Then the monomer for your carbohydrates, and sometimes they can kind of contribute to lipids in terms of glycerol, but we'll mainly be talking about the carbohydrates, is your sugar residues. And what I'd like you to notice about your sugar residues is all of the oxygen. So you've got lots of electronegative oxygens, and you have many alcohol hydroxyl groups. So lots of hydroxyl groups, and you'll also see in your sugar residues kind of an ether group on one of the corners. And so lots of oxygen, lots of hydroxyls, and an ether group there, which is kind of the backbone for the structure of your carbohydrates. And then finally, for DNA and RNA, which kind of falls into the category of nucleic acid, your monomers are your nucleic acids or your nucleobases. And so what you'll notice about your nucleobases is the element that they are very prevalent in are your nitrogens. You've also got kind of what looks like you've got some aromatic kind of things kind of going on, perchance. And you also have kind of this amine group here at the end. So those nitrogen, hydrogen things will be able to do some hydrogen bonding. You've got some double bonding, aromaticity, different things going on there as well. And so now what we're going to do, since we've kind of just looked a little bit at the snippet of the structure, we're going to go back and kind of just talk about some of the ways that these can be used in everyday life. So if we start with our carbohydrates here, and I'll kind of scroll across as we go. So again, the monomer are like our sugar residues, which in this case it's called monosaccharides. So you've got your sugar residues. I forgot this doesn't like me to write in, so I'll just keep talking. And so here's an example of where we've taken two of those sugar residues and put them together to create a carbohydrate known as lactose. And so typically our carbohydrates, those sugar molecules, are found like in pastas, breads, fruits, veggies sometimes. And what they do is they provide some of the material that's needed to build cell membranes. And they're also a quick source of energy for cells, all right? And so what they can do, the sugar molecules can kind of store energy, but then they can be broken apart, very quickly when your cell needs kind of some quick energy. And so some examples you might see are glucose, fructose, lactose, cellulose, and you'll notice a pattern of them all ending in ose, and that's kind of typical of your kind of sugars that have that ose ending. And just right quick to kind of show you, how this process of putting those individual sugars together works is typically when you're synthesizing a larger, carbohydrate, or this applies to other macromolecules as well. So when you're taking the monomer and building the polymer, the type of reaction that occurs to help that happen is what's called dehydration synthesis. And so what happens is some of the OH groups, and this is kind of where the main reaction is going to be going, some of those OH groups will kind of bond to form an ether linkage. So there's our kind of ether group here. And what will be lost or given off as a result is water. And this is why it's called dehydration, is because you're kind of removing or producing a water molecule. And synthesis, and the fact that we're creating kind of a new polymer from two original monomers. So our ether linkage here. All right. So now if we want to talk about, well, how do I maybe break them apart when I need some of that quick energy? And so in making the macromolecules, you dehydrated the molecule. We're going to kind of do the reverse effect where water is going to be on the reactant side, and it's going to be involved in breaking that ether linkage to regenerate the original monomers. And so this process is called hydrolysis, and that means that we're splitting water. So here's kind of our water molecule, and here's where it's been split back into an OH and an H. And so that's just a little bit on the reactions that kind of help us, build these macromolecules and then kind of break them down as needed. And there's more, on that if you want to read a little bit more in depth. All right, so back on to our other ones here. So we have lipids, and typically the structure of a lipid is you have kind of a glycerol backbone. And what will happen is those fatty acids, those long chains, kind of like you see here, are going to be attached to that glycerol backbone, and they'll kind of be held together there. And so typical food sources that you can find those in are like your butters, nuts, oils, things like that. You typically think of them associated like more with fats, kind of things like that. And they're good for more long-term storage of energy. They're also good for kind of cushioning, insulating our organs, and some of the materials as well can be used for cell membranes. And so some examples, like we said, are like our fats, oils, waxes are all good examples of lipids. And so kind of where they come into play in the cell membrane, like we mentioned, the phospholipid bilayer. And then we have kind of our last two categories of macromolecules are proteins, which their monomers are amino acids. And there are lots of different amino acids, like y'all kind of looked at, that all have amine groups, but they can vary, slightly in other parts of their structure. And so we have an example here of how they're using some of their structural characteristics to do some hydrogen bonding. And so some good food sources of proteins are seafood, milk, eggs, cheese. And proteins in our bodies, they help to provide structure, help us as we're doing muscle movement, and just help give us kind of good, general immunity for overall health. And so some examples of proteins are insulin, like hemoglobin in our blood, antibodies to help us fight off infections, and also enzymes that help to kind of speed up or catalyze different biochemical reactions within our body. And then finally, we have our last category, nucleic acids, which are composed of our nucleotides, or our nucleobases, as we called them earlier. So here are nucleic acids, and these are typically what you've probably seen referred to in biology classes as like A, T, G, C. It's like your adenine, thymine, guanine, cytosine, uracil, different things like that. And you'll see that they have some kind of similar, again, structures as far as, lots of nitrogen, and they have kind of a phosphate group attached there at the end that they can use as well. And so again, these nucleic acids, or these nucleotides, we don't really obtain those from a food source. Those are just, a part of us, our genetic makeup. And so again, they're what contain, our genetic information, and that's used to direct, our growth, our development, and lots of different things that occur within our bodies. And so again, the examples of those are DNA and RNA. And so what I wanted to do, just kind of as one example, is take a look at proteins a little bit more, and actually look at the idea of silk protein. And it's interesting that that little chemical molecules or structures can have a big impact on history. And so before we talk about I tell you what, we'll go ahead and talk about our silk protein, then I'll show you that picture. So one of the main ways that kind of silk has affected history is in the Silk Road, kind of way back in our past now. But we have a little picture of some, silkworms here. And so the Silk Road was kind of a big, travel pathway that they would use to pass goods. And it was named, it didn't, only pass back and forth silk, but other goods as well. But silk was one of the main, goods that was transported on kind of this different set of pathways, either by land, or by sea, kind of between different countries, China, India, Egypt, different on into Europe, and different things like that. And so the product, silk, produced by the silkworms is actually a protein. And it was, highly favored by royalty and different people of the time due to its, just very soft, silky, texture, very smooth, very, just a nice kind of high quality fabric. And so what it is silk itself. It's kind of a natural protein fiber. And it has a pattern of amino acids that kind of repeat. So like glycine, you have serine, alanine, and here's kind of a chemical structure of those things that repeat. And you'll notice that they're able to kind of connect right here with the amine groups. And so you've got kind of those peptide bonds holding them together. And what happens is, due to the nature of which amino acids are used, like the order in which they're arranged and their bonding pattern, that's what creates some of the really desirable properties of silk. And so if we kind of look at a more zoomed in picture of the silk protein, you have kind of this repeating pattern of your glycine, serine, alanine, but what happens is those can interact with other chains, the same amino acids, and they can start to link together. And so those linkages occur oftentimes by the hydrogen bonds. And again, the hydrogen bonds are able to occur because you have those oxygens, those hydrogens attached to your nitrogen. All right, kind of just label some more of those. And so what happens, what we're kind of looking at here is kind of the more in-depth structure of a protein. And so the way proteins are kind of arranged is you have your individual amino acids, and then they kind of come together to form a chain, and then those chains, again, can interact. And then when you have chains interacting with chains, those can go on and interact with more chains, and it kind of causes, all those chains to bend and take on different shapes. And so essentially like what an enzyme is in your body is kind of a bunch of these chains kind of all folded together, and the specific shape of the enzyme allows it to kind of, catch and hold or do things, to different molecules. And so when you're looking at a protein, you kind of talk about it in like 4 levels of structure. There's your primary structure, which is just kind of your basic, amino acid chain. So just like we looked at with silk, let me see. So like here would be kind of your primary structure, just your chain, your pattern of your amino acids. Then in your secondary protein structure, you have those different sequences of amino acids kind of linked or connected by hydrogen bonds. And those can either take kind of a helical shape, which is called an alpha helix, or it can kind of look like more of a zigzagged sheet, which is called a beta pleated sheet. And then what happens when you take those beta sheets and alpha helices and kind of, get them interacting with each other, that create what's called a tertiary protein structure, where you have interactions between those different sheets and helices. And then finally, kind of the functional unit of your protein is what's called the quaternary protein structure. It's kind of level 4, and that's where you have more than one of these amino acid chains that kind of come together, again, take on a specific shape. And so that's what allows things like, hemoglobin to carry heme groups, to perform the function that they do in our blood. And just, those shapes, the quaternary protein structure really determines what a protein is able to do in our body. And so kind of an example of that quaternary protein structure is here. This is actually some quaternary structures of spider silk. So, not exactly the same as silkworms, but related. And what they typically do, because these protein structures can contain, so many amino acids, they tend to kind of just smooth everything out. That way you just see kind of an overall picture of like the different helices, the different, pleated sheets, but they don't necessarily show like all the chemical names. And they'll oftentimes use abbreviations like for your amino acids. So like instead of glycine, put G, something like that as well. Because there are just so many of them that are involved in these different proteins. All right, but again, it's okay if all of that didn't make complete sense. We're just exposing you that, hey, this is cool, this is amazing, we're fearfully and wonderfully made, and that this stuff is out there if you want to go and learn more about it. But again, kind of the main takeaway from today is that chemical structure affects function. All right, and that holds true in biochemistry and in just lots of other areas as well. All right, well guys, thanks so much for watching, and we'll see you next time.